Wonderful. Good evening or good afternoon, everyone. Welcome to the BACE event on epigenetics of the microbiome. I'm very excited for our session tonight. For those of you who are new and for everyone, I just wanted to remind you that the mission of BACE is to bring together diverse professionals with interest in epigenomics to help us all learn, share, and help elicit opportunities in epigenomics. We do that via typically bi-monthly events, and every other month we meet for so-called chromatin coffees where we interact in a bit more informal setting. And you can find more information on our website. Without further delay, I'm very happy to announce our agenda today. We will soon hear from Gabriela on the first talk of the night. We will have a brief Q&A afterwards, then we'll hear from Ivan and we'll have another Q&A, and then we can actually spend a little bit more time with both of them afterwards, chatting in breakout rooms. As I mentioned, the talks will be recorded, and if you would like to participate in Q&A, let us please put your questions in the chat, and that way I can hopefully help pass them to the speakers as we go along. So without any further ado, I'm very happy to introduce our first speaker tonight, Gabriela Gutierrez, who is the founder and CEO of Microgenesis. And she will tell us about the gut health and the fertility biome. Gabriela, welcome. Thank you so much for joining us tonight, and the floor is yours.
Thank you, Monica, for this nice opportunity to share our science and our results with you today. I like to share my screen now. Please let me know if you can see it and how it works. I like a few videos to show you in the background when I'm talking to you. Please feel free to share your question or interrupting me anytime. This is not a formal talk, so you can interrupt me. I am from Argentina. I am from Buenos Aires. I'm a scientific researcher from CONICET, our National Agency of Science in Argentina. I spent the last 15 years working in the field of infertility, trying to connect, trying to bring more diagnosis to this field. Actually, I like to share with you the mainly problem with infertility is two mainly problems. One is about the diagnosis, and second is about the innovation on this field. The diagnosis, you need to know that fertility treatment is controlled by IVF centers. In vitro fertilization technology is more than a 30-year-old technology. And in the last 13 years, we don't have a lot of innovation in the field. IVF, in vitro fertilization, is a treatment, is not a diagnosis. And everyone in the world can see that it's a lack of diagnosis in the field. Any woman from any country spend in average at least 38 months to get a diagnosis of infertility. And that apply only for the ones that can afford a doctor service to realize that they are infertile. So it's not only about the time they spend to realize that they need an IVF procedure, but there is also that only 10% of women can afford a fertility treatment in the world. So our science and our technology have the mission to change that, to democratize the treatment for infertility, but also to bring some innovation in the diagnosis field to short the time that people need to realize that something is not going very well on that. So let me tell you about our fundamental insight. Sorry about that. In Microgenesis, we develop not only a test, but a treatment that links for the first time a gut condition, a microbiome condition with infertility. We spent 15 years working on inflammatory subclinical factors that affect the fertility potential. And during the last 4 years, we realized that those inflammatory factors are linked to our microbiome, our gut microbiome. When functional microorganisms are missing, we call these functional microorganisms from our gut microbiome fertility biome, the ones that are linked to the fertility potential. When that microorganism, bacteria and fungi and any other microorganism that take a very important role on the prevention on inflammatory conditions, but also on the reproductive potential, control, for example, the functionality of the ovaries, the functionality of our pancreas, the functionality of our thyroid. When these microorganisms are missing, this creates a specific disbalance that show our immune cells that are in the intestinal barrier a specific molecular pattern, and these immune cells at the intestinal barrier secrete a specific microRNA signature. These microRNAs are the functional markers that connects the microbiome disbalance with a specific inflammatory condition. For example, a specific microRNA that is secreted as a consequence of a disbalance between yeast and bacteria create a specific microRNA that control the integrity of the intestinal barrier, creating, for example, a leaky gut condition. And in time, with according the process is going on, these microRNAs control genetic expression. So they get into our bloodstream and they move to every single cell in our body, so it can produce chronic anemia, it can produce chronic hypovitaminosis, but they can also affect our metabolic pathways or create an autoimmune conditions. So we spent the last 15 years working, connecting these subclinical inflammatory markers we get from a blood test and a saliva test. For example, insulin levels, cholesterol, autoantibody levels. We evaluate 8 different metabolic pathways that are associated with a specific gut condition that affect the fertility potential. So we created a test. We deliver a test box to patients. Let me show you in the video that we deliver a test box to patients. This test box contained 3 different sample bags. One sample bag that have a tube to collect a split sample, one DBS, a drop blood spot, a lancet, and a DBS card when you can squeeze a drop blood and breathe it out. And we analyze the blood markers through a mass spec analysis. And after 10 days, we deliver a specific report telling you about what is going on your microbiome balance, but also how it is impacting your 8 different immunometabolic pathways associated with that disbalance. According this microRNA and blood and saliva tests, we are able to recognize 54 different profiles. And we associated this profile with a fertility plan, a restoration plan, combining meal plans, customized nutraceuticals, and customized blendings of probiotics according to the result. Let me tell you what these 3 components are important for this diagnosis. or to treat this inflammatory condition that affect the fertility potential. This is because we use 3 preparatory blendings of probiotics. We combine strains of probiotics you can't find over the counter to replace this missing key microorganism. In order to allow the probiotics to grow in your gut, we offer a meal plan to bring the prebiotics that these probiotics will need, but also to create an educational program to select the food components that will help you and to avoid the ones that will provide or will favor the growth of the microorganisms that are overgrowth in response to the ones that are missing. So it's not only which probiotics should I need, but also which functional food or fermented food we need to avoid. And this is a 75-day plan, meal plan and restoration plan. And the third component about the nutraceutical, for example, we use omega-3, resveratrol, glutamine, selenium. We use this. We spent 15 years trying to replace traditional immunomodulatory drugs such as steroids for these nutraceuticals. We are very aware that women are not sick. You need at least 10 years to develop an inflammatory process to become a specific inflammatory condition, a specific inflammatory disease, for example, Hashimoto disease. But in the meantime, in this time frame of 10 years, when women trying to get pregnant, this subclinical process is not a specific condition, but is in the way to create an autoimmune disease or a metabolic syndrome or endometriosis. So we replace the traditional drugs or prescription drugs for nutraceuticals. So this is a food supplement restoration planning. And this is based on our science we publish in our article. We ran a clinical study including 40 fertility clinics from Spain and Argentina. Spain is the second biggest fertility market in the world. So we spent the last 3 years running our clinical study there. And we included 287 women that failed at least 4 in vitro fertilization before being included in our study. We selected these 4 failed IVF procedures because IVF success, it's about 30%. So every woman in the world needs at least 3 IVF attempts to get pregnant. So that's why we included the woman to this study after failing the 4th attempt. So there were multiple IVF failure women, 40 years old age, non-baby born in this group before being included in our study. And after we tested them, after they finished our 90-day nutraceutical treatment, 75% of them got pregnant within 6 months. 70% of them got pregnant in the next attempt of IVF, but 30% of them got pregnant spontaneously in the meantime. So we find the way to improve the fertility outcome, even for the ones that already failed IVF procedures. As I mentioned, this is published in the American Journal of Reproductive Immunology. And we are trying to apply this technology for 3 different segments. The ones that are failing IVF procedures, the ones that are before trying to conceive, trying to conceive naturally with their OBGYNs, but in the future, our vision is to bring a tool for women that are dreaming to have a baby, and they want to have a tool that bring information about the fertility potential. And it's not any test in the market other than the one that is measuring ovarian hormones. So after we finish our first clinical study, we started an alpha study in the US. We wanted to repeat the proof of concept here. And we invited 40 alpha users to our program early this year. 23% of them are already in the pregnancy planning. They finished the 75 days restoration plan. In the first month, 3 of them got pregnant. And in the second month, 5 of them got pregnant. So we moved the pregnancy rate, the 5% pregnancy rate they had in 35 months of seeking for pregnancy period to 35% in only 2 months. So it's not only we can improve pregnancy rate, but we also significantly reduce the time they are struggling with infertility. So we already demonstrate that we decrease the upregulated microRNAs with our customized food supplement treatment. We reduce intestinal permeability. We improve the overall health and wellness of our patient we get from our testimonial, and we increase the fertility potential. Microgenesis technology is not only applicable to women, it's not only applicable to fertility, but we are planning to use the same platform to test men. We are just about to start a male partner test. We want to demonstrate that microRNAs can be exchanged, kissing each other's. They can find in every single human fluid. And we want to demonstrate how your microRNA is affecting the fertility potential of your partner. And we want to treat both. But in the future, we want also to prevent early onset diseases in infants, such as celiac disease, creating a personalized meal plan and nutraceutical supplementation for the entire family. Thank you very much.
Thank you, Gabriela. That was really impressive. Are there any questions? I don't see anything in the chat. Please feel free to post in chat, or I heard Gabriela say that she doesn't mind you speaking up and asking directly. I can kick off. I'm really excited. I wondered if, and I don't know if you can share that with us, but are there any microbial species that seem to be the most important in the mix, so to say?
Yes, we were able to realize that it's not only about how much lactobacillus. Everybody knows that lactobacillus are very important in the vaginal microbiome. We were able to demonstrate that your gut microbiome is connected with your vaginal microbiome through these microRNAs. And it's not only about how much lactobacillus you have in your reproductive tract, but also the species that is dominant. So it's not only about supplement lactobacillus, is to select the specific species that you need.
Interesting, thank you. We have a question in the chat. Could be that would the fertility dysfunctions due to the activations of the immune system be related, for example, in the event of blastocyst implantation?
Absolutely, but is the immunological factor that is created after the microbiome disbalance that is affecting the fertility outcome. So for reproductive immunologists, at the beginning was, okay, inflammation is affecting fertility outcome. Now we know that the real problem is start in the gut. So first is the dysbiosis, then is the immunological factor, inflammation, and that is affecting the fertility potential. So it will not enough to just control inflammation. We need to restore the gut issue to prevent the inflammation to progress in time.
And we have another question. The vaginal microbiome secretes microRNA that inhibit pregnancy. If so, then why do you think this is? I'm sorry.
I understand the question.
Oh, do they secrete microRNA? Right.
Yeah, yeah. Well, the microRNA we test for is secreted by human cells in response to specific bacteria, right? So it's like an antibody. You can create a specific antibody against microorganisms. The same happens with microRNAs. So you have a specific microRNA disbalance, so you will have a specific microRNA signature. And this microRNA will be affecting, controlling, epigenetically controlling all your metabolic and immunological response. And this will control any other microbiome. That's why, for example, psoriasis and chronic candidiasis is associated with gut health. Is that answering your question? You're welcome. Yeah, this is a self-collection test. It's a lancet and a tube and a vaginal swab, very easy to collect. You can ship these samples to our CLIA lab in California, and we ship a restoration kit containing the personalized nutraceuticals according to the tests and an e-report containing the meal plan for 75 days.
Great, thank you, Gabriela.
Thank you.
Looking for more questions. I think we have time for one or two more. I wondered if you, and maybe you mentioned that, would you be able to share with us a little bit about the molecular principle of your test? How you are able to assess the microRNA?
I'm sorry, can you repeat the question?
Oh, I was just wondering, since we have time for maybe one more question,
Yeah,
I wondered if you could elucidate a little bit about the molecular basis of your test.
Yeah,
how do you assess the microRNAs? Thank you.
You're welcome. We're using PCR to assess for microRNAs. MicroRNAs can target different RNAs. One single molecule of microRNA can target with different affinity 100 different type of RNAs, of messenger RNAs, and they control genetic expression. They can silence this specific RNA. So that's why they are controlling gene expression. So they are the connection between our lifestyle, our food habits, and our inflammatory conditions. And we are the first microbiome company that is not using NGS, unit generation sequences analysis, to test bacterial DNA to look for specific microRNA disbalance. And that is why we did that for our clinical study. And after that, we nailed the results with a specific microRNA signature through a PCR test. A PCR test makes the technology faster and cheaper, and more scalable for every single lab in the world after COVID.
Wonderful. Thank you, Gabriela. I see one more question asking whether this is also for people that will have IVF.
Yes, as I mentioned, we are trying to target 3 different customers: the women that already are failing IVF procedures, women that are before IVF, they are just trying to think how I will avoid IVF, because some of them can afford IVF. 90% of infertile women can't afford IVF treatment, but some of them just want to have less invasive treatment, or at least a diagnosis before getting into a fertility program. This is our first objective, to democratize the access to a treatment, but also to bring a natural solution before IVF.
Wonderful, thank you. I don't see any further questions. If that's all for now, then I think maybe we can move on to the next speaker. Thank you so much, Gabriela.